Hello everyone, I'm Dr. Jim Daly from Daly Manufacturing, here again with you this month for our November 2019 monthly training. Today we're going to be talking a little bit about herbal extracts. We use a lot of these in our industry, and for good reason, and we'll go into those. Traditional uses of herbs go back thousands of years. Herbs have been used for foods, medicines, for lucky charms, and used in religious ceremonies, many different ways for many, many years. Now, the early Native Americans used herbs, but they used them not as extracts really. Well, yes, they were extracts, and a water extract, that's what a decoction really is. But they boil a big handful of roots and leaves and whatever in water. And then a sick person or whatever would drink large quantities, cups of that water. And that's how they would do it. It's a rather cumbersome method for us today, but it worked well for the Native Americans. And the Native Americans were very well adapted for their environment. They could live quite well wherever they were. In fact, I read an account by an Englishman who, in the very early days of the Americas, spent some time with the Cherokees, and he was amazed. He said you could put one of the Native American Cherokees, Indians, out in woods with nothing, and it wouldn't be long till they'd made tools from rocks, made a little bark hut, had a fire going, and was gathering food to eat. So they could pretty well survive and live well just about anywhere. And part of what they had adapted to is learning about plants and other things around them that could be used for medicines. Their method was probably very good for a lot of the compounds in your herbs, particularly your water-soluble ones. Oil-soluble ones, they probably didn't get as much out. You'd get some out if you boiled the water, but a lot of those were probably wasted by those methods. And some compounds were probably destroyed by heating or perhaps volatilizing and went off with the steam. So you didn't get everything out of the herbs, but they knew which ones they would be able to get something worthwhile from by boiling the water. However, herbs were not always available. If you might have a favorite herb, and it might be out of season if you wanted to use it. Nowadays, we still collect herbs like they did, but we're a little bit better at saving them and preserving them, perhaps, although the Indians did do that. In fact, dried herbs are often stored for quite a long time. So we don't today have to be limited to what we can grow ourselves or collect locally. We can often buy herbs from local stores. We can order almost anything over the internet. Herb shops often have a lot of herbal medicines. That's a picture of one, I think, in Korea, where you see this quite commonly there, actually. People have shops like this, and you'll see, baskets of different roots and barks and leaves and whatever that people will buy and make their own medicines. However, that's not really practical for a lot of people either. It is also an inefficient use of plant resources, since many of the bioactive compounds are just not really made available by the methods that people use themselves a lot of times. Now, we have two different types of approaches to healing: the drug approach and what we call the natural medicine approach. And they really focus on something a little bit different. The drug approach to disease mainly manages a single target, an enzyme or something like that can be modulated by a chemical compound. For example, the cyclooxygenase 2 inhibitors such as Siloxib It's a very strong inhibitor of cyclooxygenase 2, and it is effective for pain. But it is so strong it also has a lot of side effects. Um, so we find that, Celebrex, which you're familiar with, is no longer on the market because of the side effects of it. It works in a way too well. Traditional Chinese medicine takes an opposite approach, uh, using herbs or less potent extracts. Sometimes I've wondered if their products could be effective with such low potency, actually. However, they often use numerous low potency ingredients. And what I would say here, in a way, the drug approach is more simplistic in a way and less complicated than the traditional medicine, because if you're using something to inhibit cyclooxygenase 2, well, you kind of know exactly what it's doing. It may do it too well in that case. But the same thing's true like with statins. You know that they inhibit HMG-CoA reductase, the rate limiting step in the synthesis of cholesterol in the liver. But that same step that inhibits the manufacture of cholesterol in the liver also inhibits the manufacture of coenzyme Q10 in the liver and a lot of other things. And that's one reason why people who are on statins really need to take coenzyme Q10, because their body doesn't make it like it should, because they are inhibiting a step in that process. However, when we look at traditional Chinese medicine and other natural healing processes, they use things that do it much less potently, but still effectively. But also, a lot of times, they're things that will attack the same problem from many different angles. And we'll talk about that a little bit more, but that can also work for drugs. This is a study about 2.5 years ago now that I've mentioned before, but this was using actually drugs that are very powerful inhibitors like the other two we've mentioned. However, what they did was to give 1/4 of the lowest effective dose of 4 different drugs for hypertension. So the lowest effective doses you could get, they had to have somebody actually make it for them because you couldn't buy it in that low of dose. They took 1/4 of that dose and blended it for 4 other ones. And they found that it actually worked just as well or better than any of the other drugs at the full potency, and had much less side effects. But this had attacked it from different things. There was a ACE inhibitor in there, a calcium channel blocker. I forget what the others two were, but each one dealt with the problem at a different angle and from a different way and had a very small effect. But the combined effect of all 4 of them was quite good, and the safety profile was much better. None of the patients had any of the side effects you would expect from the hypertension drugs. So that shows that this type of method works, and actually it gives me a lot of confidence in the natural approach to healing. Now, sometimes we take the drug approach with natural products as well. We have compounds that are almost, pure compounds that are extracted from drugs. Mucuna is 90%. L-DOPA is available. Grifonia simplicifolia is 99% 5-hydroxytryptophan, the precursor to serotonin that you use in the brain. You can get green teas with 99% caffeine, theanine, and 98% caffeine. The Polygonum cuspidatum, resveratrol, can be purified as much as 98%. So here we're looking at pure compounds in a way, even though they're extracted um, from plants, but they really come down to pure chemicals. So when you use something like that, you're still using kind of a drug approach to healing and not so much as of the natural approach. But sometimes it's effective, and sometimes you can combine these together with other things too. And you may need to do that occasionally to get the dose up to where you want it. Uh, Now let's talk about one of the things that we deal with quite often uh, is pain. And the most important pain receptor is called TRPV1. Um, that's transient receptor protein valenoid 1. So just to be more specific, and this is where a lot of the pain we have to deal with begins. And it's at the nerve ending. So if you pinch your hand, well, you're activating that TRPV1 receptor and you feel a little bit of pain. It can also be stimulated by heat and cold and chemical burns and a lot of different things, and gives that kind of burning sensation. Many TRPV1 receptors are at the skin surface, but some are also located internally, such as the ones that are involved with joint pain. Now, valenoids, and we'll go over this pretty quick, is a big category of compounds that come from plants. The most simple one is vanillin from the vanilla beans. I've never yet seen it be used for treating pain. I'm not sure it does well. It may be you need that side chain on it to work well, but I've never seen anybody that's tried it one way or the other, so I've always been curious whether it would. Capsaicin is something that's commonly used for treating pain. It's used in a lot of pain over-the-counter medicines. Gingerol also is a valenoid and used for treating pain. All of those, however, work on that TRPV1 receptor. And the way they do it is by desensitizing it and making it less able to cause pain. And exactly how that works is a mystery. Honestly, the people are still trying to figure that one out. But in some way, these pain-causing substances can cause the TRPV1 receptor to not be sensitized and not react to the pain in some cases. However, I've never been able to see that putting very hot red pepper in your mouth, it always is hot, it seems like to me. So, it doesn't seem to work everywhere, but particularly in some of the joints and things, capsaicin seems to desensitize the TRPV1 in the joints. So there may be slightly different receptors for some TRPV1 molecules in the body that work better than others. And the funny thing about it is, and we'll just mention briefly, that the effectiveness of red pepper is really not totally dependent on the hotness. So it's not that you're just burning it out. These molecules, there's some analogs of capsaicin, which is hot to your mouth, that are not hot to your mouth in the analog form, like the capsaicin is. But they still have the effect of reducing pain, and also some other effects that capsaicin and its analogs have, particularly metabolic effects. So it's interesting that it's not just the hotness and the pain that makes it work. It's actually the way the molecule binds to the receptor, the TRPV1 receptor. Now, there's multiple sites and multiple products that really can work on these pain receptors and take it from different angles, just like we were talking about with that blood pressure medicine where they used the 4 different ones. Several things act directly on pain receptors. although in a somewhat different way at sometimes. Glucosamine chondroitin actually seems to help relieve the pain in the joints, not so much by acting on the receptor, but by improving where the damage is in the joint itself. So you're not really, it's not a pain blocker, but getting to the root of the problem in that case, which is the preventing the pain. There are inflammatory targets that we can do. Inflammation actually highly sensitizes the pain receptor and makes things affected a lot more. So we look at things like tumor necrosis factor alpha, NF-kappa B, which is a major regulator of inflammation. The pro-inflammatory eicosanoids, like that's one of them was the COX-2 we were talking about earlier. Interleukin-1 is a immune factor that stimulates inflammation. Well, there's a lot of things that will interfere with that inflammation process, and by extension, the pain receptor. The omega-3 fatty acids do that. They work a little bit different from a normal inhibitor. They actually replace the lipids in the cell wall that are made into the pro-inflammatory eicosanoids with something that doesn't make a pro-inflammatory eicosanoid. So it doesn't really inhibit it, it just replaces what it needs to do it. Curcumin, boswellia, tea, cocoa, these all are inhibitors of the inflammatory process. Quercetin again, sulfur compounds like MSM and garlic. These are all inhibitors of inflammation and will reduce pain, but from slightly different ways. Red pepper and ginger are ones that act directly themselves onto the pain receptor. And then you get into the cannabinoids and opioid receptors, which actually block the signal from TRPV1 to the brain. And we have natural products that do that. Kava kava, green tea, and cannabinoids, and of course the opioids themselves do it. So in that case, you have the pain at the pain receptor, but you don't ever feel it. You know, the signal doesn't get through to the brain. So, you know, you've got the pain, but you don't care. It's what it really amounts to. Well, all of these products, let me go back. All of these products, we almost always use them. If you look at omega-3 fatty acids, that's really an oil extract from fish. Curcumin is always extracted from turmeric. Boswellia is an extract. All of these are extracts of one kind or another. The ginger and red pepper can actually be used if you have fairly high doses without making an extract. The whole plant can be effective. However, kava tava, green tea, probably be effective. You need a little bit more of an extract to get there. So only a handful of herbs can be used directly, effectively, without being extracted, unless you want to use a very big doses like we were talking about the Native Americans doing, where they make this great big pot of tea and throw in handfuls of herbs and boil it and drink, you know, cups and cups of the decoction. So we look at different types of extractions, and that's what we're going to talk about for a little bit now. There are lots of ways of making extracts. First of all, you've got to deal with solvents. Whenever you make an extract, you pull the bioactive compounds out of the plant and into a solvent of some kind that you can then concentrate. Water is probably the one that we have used over the years the most and the longest. And it's still used a lot. There's a lot of things, if you're wanting to make extracts, that you can actually do with just water as a solvent. We get quite a few herbs in that use just water. it's also the one that is the most accepted by consumers. They, pretty much assume that the water's not going to harm them. Then you get carbon dioxide, supercritical extraction. It's a quite expensive process, but it's also one that's very clean. Carbon dioxide they release it into the air, which you may not like too much, but compared to the other other sources of carbon dioxide is pretty small. But essentially, they put it under pressure and cool it down and make carbon dioxide into a liquid in which they extract herbs. It's like I say, a very expensive process and involved, but very clean process. You don't have any residues of any chemicals. Ethanol is the other one that's fairly common in this industry. It's considered to be relatively safe. In the content, the amounts that you would be able to get in a dry extract are insignificant. So, it's not like you're going to be drinking alcohol. But it can work very well. It can help get out some of the more oil-soluble things that you really have a hard time with water. Acetic acid or vinegar can be used as a solvent. Methanol you see sometimes. People don't like that as much. And in fact, in Korea and China, they don't allow you to use methanol in extractions. People can here in this country, and actually the methanol in the very, very tiny amounts is not toxic. Your body can handle it and metabolize it. It's when you get higher levels of it. And your body does it all the time. It's actually something that's just in our food, and actually our body makes it under certain circumstances, and it has methods to deal with it. So very tiny amounts are not problematic with methanol, but it's still not well accepted by people, and it's illegal in a lot of places. Then you get to the acetone and hexanes, which very rarely show up in any kind of food product because it's really not acceptable. There may be some things that would be very hard to get out without using them. And I used it for extraction, for food extraction, just for testing, because it got the fats out very efficiently. And I could actually efficiently enough, I could quantify the amount of fat in a food with that kind of extraction. Whereas you wouldn't get it out as completely probably using any of the other things. So for that it worked. But for food preparation or dietary supplements, not really acceptable. Now there's other things that we also use. There's methods besides the solvents. Frequently use fairly high temperatures, and there's times we don't want to do that. But for some things that it's quite acceptable. Pressure is used to help get the bioactive compounds out. We use a reflux method, what we call a oxalate extraction, that helps speed up the process, and I'll show you that in a minute. Also, just soaking for a long period of time in solvents without any particular treatment other than just the soaking works well. Fermentation can sometimes get things out, sometimes actually modify them a little bit. And an interesting thing that has been published a few times lately is just micronization of the whole herb. It appears that if you grind it into a very small nano-sized particle, that you really, in the digestive tract, get those things out a lot more efficiently. And so you can get some of the benefit of extraction just by finely grinding a whole herb. And that's not something that I've done yet here, but it's not well enough studied yet to know which herbs that will work for, and getting the doses right. It's not a traditional method of doing it, and so we don't really know too well yet how to make that work right, but we do know that it's got potential. Well, this will just tell you kind of the compounds that work for different solvents. Water, you can get out starches, tannins, saponins, and the saponins are, you don't think about them too much, but you know, like ginseng, the saponins are the active ingredients in ginseng. So a water extract would work well for that. Terpenoids will come out with that. It's a very valuable set of compounds. Polypeptides, an interesting group we don't know enough about yet, and lectins will all come out with just a water extraction. Ethanol, uh, well, you can get your tannins, polyphenols, polyacetylenes, flavonols, terpenoids, alkaloids, and sterols with ethanol. So a lot of different things come out with ethanol. Supercritical extraction is used a lot for essential oils. Most of those are made with the CO2 supercritical extraction. The phytosterols are often made with that as well. And anthocyanins are a tough one to get out. Methanol will do it, and probably one of the few things you might use methanol for is to get out the anthocyanins and concentrate them. They may have other methods to do that too, but you know, that's one that really is hard to get extracted with the other methods. Now this is an illustration of the Soxhlet method just to show you what it does. And essentially what it does is keeps a fresh solution of your solvent working in the plant material that you're trying to extract. And if you look down at the bottom, you see there's a heated flask down there. And that's the solvent. It could be ethanol, for instance, that's being boiled. The steam comes up, it goes up all the way to the top there where you see that kind of funny looking thing, looks like sausage or something. Well, that's a cooling chamber. You see the cool water goes in at the top and comes out, in the bottom and comes out the top. So it cools the solvent down. It then turns to a liquid, drips down into that test tube that contains the substance that you're trying to extract. Now you'll see that as that fills up, it will finally, on the left there, go all the way up and suddenly siphons out, and it goes back down into the flask with all the stuff that it's extracted from the plant. Then that solvent is just simply recirculated. It volatilizes again, goes up, cools down, and goes back in there. So you keep putting fresh solvent into that flask to do the extraction. And that really speeds up the process quite a bit. And then when you're finished, you take that flask down at the bottom, use some method, probably a rotary evaporator if it's that size of one, to evaporate out the solvent, and you've got your extract. Very neat way of doing it. That's something I used to do in the lab at the University of Tennessee when I was there. The supercritical extraction, and I pulled this off of eBay. They actually had a small pilot model supercritical extractor for sale on eBay for $378,000. So, and that's not even what you'd use in a production size operation. So you can see that you can get into some serious money with that technology. But like all extractors, it uses a solvent to diffuse, for the ingredients to diffuse into. The pressurized CO2 gas forms a liquid, and the CO2 solute goes in through a series of depressurization vessels where the extract remains as a liquid and the CO2 exits as a gas. That's a very short way of describing it, but it works very well for a lot of things. Now, when we make the extracts, we make them different ways, and you'll see this on a lot of bottles of herbs and things. You'll see it described in different ways. You'll see maybe a 4-to-1 extract. What that means is that 4 kilograms of herbal powder was used to make 1 kilogram of dried extract. That's not extracted out very much, but it has probably gotten out most of the cellulose. Also, you have a 10-to-1. There again, a 10 kilograms of dried powder herb makes 1 kilogram of dried extract. Well, these are not standardized to one thing. And ideally, you would have all the bioactive compounds, and there can be dozens to hundreds in an herb, still there. And they often have the potential to act together synergistically. The particular herbal extracts and plants that have kind of risen to the top, I think, to be more effective, most of them probably do have multiple compounds in them that actually work together. And that's something we don't know enough about, is the interactions of the different compounds in the herbs, honestly. It's very complicated. There's a lot of different herbs with a lot of different compounds. And how all these combinations work, we don't know. But we do know that apparently they do. Now, more often now, we're seeing standardized extracts, where the extracts are concentrated just like with the other ones, but then to a state where you have a known amount of one compound that's considered the primary bioactive compounds. When it's done right, those other compounds will still be there, at least to some extent, but it will favor, their method will favor that one type of compound to really concentrate it and bring it up. And we see a lot of things like that. You'll see silymarin is an 80% extract usually from milk thistle. So you have it concentrated to a very high concentration of silymarin, but you still got 20% of plant material there, compounds that are in there that are not silymarin. And to what extent they are helpful in making silymarin more effective is not known. But we do know that oftentimes they've done studies with comparing herbal extracts with just the pure compound made chemically, and it hasn't worked as well with just the pure compound. That's true with St. John's wort and hypericin. Hypericin is typically, at least used to be, considered the main bioactive compound. That's what it's standardized for. But when they've done tests comparing the standardized extracts of St. John's wort with just pure hypericin, it doesn't work. The hypericin doesn't have the effect on depression, for instance, that the St. John wort extract does. So we know that these things work together. And ideally, they will have at least some of the other compounds in them. And some more examples of herbal extracts standardized to a fixed compound of active ingredient. Often it's one, sometimes it's multiple ones, like ginkgo biloba is standardized to flavonoid glycosides, usually 20-some%. The ginkgolide, A, B, and C can be up to 4, 5, 6%. Bilobalide, there again, 3 to 6%. Terpene lactones, 5 to 12%. But all these things in ginkgo sometimes are standardized to those multiple things like that. Tricky to do, actually, to make it that way. But that's the way they do it to have the most effective ginkgo extract. And as we said, the 80% silymarin. Echinacea, common one is 4% chicoraic acid. Astragaloside 4, they make up to 98%, and curcumin 98%. So you can have a range of extract concentrations in these standardized extracts. Extracts have the advantage of producing the standardized extract a known amount of an active component. So if you're getting that milk thistle with 80% silymarin, you know exactly how much of that active component you're getting all the time. If you drink milk thistle tea made up like the Indians did, well, you don't know how much you're getting because it would depend a lot on the conditions when they harvested it, how long they boiled it, things like that. So you don't really know your dose. With the standard extract, you can have a dose and you can have it the same every time and know exactly how you're doing it. But the disadvantages are, anytime you concentrate one thing, you know that you're removing something else. And it's always possible that you're removing something that you really would like to have. And sometimes we just don't know all the compounds that are really bioactive in herbs. So we do the best we can, and we concentrate. And I like to use standardized herbs in combination with whole herbs a lot of times. You'll see several of ours. If you look at our milk thistle, we use the 80% silymarin, but then we also put in whole milk thistle powder with it instead of using microcrystalline cellulose as a filler to fill up the capsule. And that way, you know, you're getting a little bit more of some of those other things perhaps that you might have missed. Now, there are different ways of concentrating it, and a lot of those are secrets that these companies have. They don't really tell you how they do it, to be honest. But a lot of times you have to be able to increase the amount of bioactive compound to the desired concentration. The extraction methods that we use, the crude extractions, just kind of extract everything down and doesn't usually separate out the one compound. That can be separated by an ultrafiltration type process, membrane filtration, dialysis. The high performance liquid chromatography that they use for assays can also be used in what they call a preparative chromatography to remove particular compounds. You would usually only do that commercially if you had a very expensive compound you were trying to get out, because it is an expensive way of doing it. But something like extraglycide 4 could be done that way because it takes, many pounds of herb just to get a little bit of that extraglycide 4, and it's very expensive. So, that would make sense. Well, we talked a little bit about the, and I was just going to mention one more thing about that, TRPV1 receptor. I think we'll quit after this slide. The TRPV1 receptor, doing things that can suppress that, really does have some major health benefits. Knockout mice without TRPV1 at all had better insulin secretion. They had increased energy expenditure. They were leaner. They lived longer and had better blood glucose regulation. That's when they made transgenic mice where they didn't have that TRPV1 at all. Now, do we want to replicate that in people? Not really. Reason being, people have been born without TRPV1 receptors that were active. They don't live long because, despite the health benefits of having that gone, they also don't feel pain. So there's nothing to keep them from doing dangerous things. They don't have the feedback. If they burn themselves, they don't know it. If they cut their leg, they could bleed to death before they realized that they had even done it. They tend to injure themselves, engage in very risky behaviors just because they can, and they don't have any consequences that they know of immediately. So it's really not a good thing to not feel pain. However, when we can suppress that, we actually do see besides the pain benefits, the same benefits that the mice felt when they didn't have the pain receptor, but on a lesser level. Suppressing pain really does help your body. And a lot of people say, well, why do you want to suppress pain? Why don't you get at the root cause of the pain? But in a sense, that's not a quite true analogy. The thing about pain is these things that we talk about, pain, it actually feeds on itself. Pain activates that receptor, which has a cascade effect and has a feedback that causes more pain. So you have to break that cycle at some point, the pain, inflammation, and then more injury. The pain itself causes injury to the tissues where we are. So the inflammation causes injury to the tissues. So you have to be able to break that cycle, and that's why we treat the pain and why we want to look at it from that angle. And these herbs that we have, curcumin, ginger, these are wonderful substances, the capsaicinoids, to help you reduce pain and help you reduce some of the effects of the TRPV1 activation. And we know that those things like gingerol, capsaicinoids, they do improve insulin secretion. There's been several studies on that. We don't know if that makes you live longer, but it does improve glucose regulation. So these are things, when we think about treating pain, we're really treating a lot more than pain. We're really helping the whole body. And that's what these herbs do in these extracts. They are often taken for just one thing, treating pain or whatever, but they can also do many other things. And the extracts, we keep learning more about them, but I hope this today at least help you understand a little bit the process of making extracts, why we make extracts, and why we like to use them. And I thank you for joining us, and have a wonderful next 4 weeks, and we'll be back again for another topic. Thank you very much.